Can we save or restore ʻŌhiʻa? The first question is, how bad will it get? The other thing is how strong is public support for a program, because nowadays nothing's cheap, including a resistance program. Some of the fundamental questions that come up are, what level of natural resistance do the species have? How do we harness that natural resistance in an efficient and timely manner for restoration? Can we retain the genetic variation and genetic integrity of the species? And then, have resistance programs succeeded in any other species? And yeah, the answer to that is yes. It's a qualified yes. And some people equate resistance with immunity. And I'll tell you right now, you're probably not going to get to the stage where you have a diverse population of ʻŌhiʻa that 100% of the seedlings that you plant will survive. That doesn't work with any of the other programs unless you go to major gene resistance, and with major gene resistance, that's going to be overcome in a few years, just like it is in the crops. In some cases, resistance may be the only feasible solution. You want to exclude new pests or pathogens, okay? You try to do that, but unfortunately they come in. Then you want to eradicate or confine the slower spread. And you can try that, but in some cases that doesn't work. Some cases you might try biocontrol. In many cases that doesn't work. Silvicultural management, that may help. Monitoring certainly is good, but as I tell people with tan oak, I don't care about monitoring if that's all you're going to do. I don't want to know when the last tan oak is going to die. I want to know what we're going to do about it. And the neat thing about genetic resistance, at least the natural genetic resistance, is hey, it's organic, it's generally sustainable, and it usually has little or no side effects. What is a breeding program? It's an iterative process of selection, mating, testing. It's been going on for thousands of years. The goal of producing improved populations of parent trees for whatever objective you want. What tree breeding isn't, and sometimes there's confusion out there, it's not transgenics, it's not gene editing, it's not identification of resistance mechanisms, it's not genomics, transcriptomics, proteomics. Those are tools, but they're not required for successful resistance program. As a matter of fact, in tree species, probably, there's a paper that Jennifer Cook and I wrote 2 years ago or whatever it was, and all the successful programs to date have not used any of those tools. Now, that's not to say that those tools will not be useful, will not make things more efficient in the future, but they generally haven't been right for prime time, and you still have to have identified resistance to begin with before you can use those tools. Genetic variation is a cornerstone to evolution of a species in the face of almost any abiotic or biotic event that comes in. Phase one, before you decide whether you need a tree breeding program or resistance to, so that you can get all the models correct and everything too, is you have to ask, is there resistance? What is the frequency and distribution of resistance? What type of resistance? Because there's different types of resistance. Some of them will be ephemeral and not last very long. Some of them will last hopefully forever. What level of resistance, or what I call the usability? But then you also have to ask, I tell people for tree species, unlike crops where they plant each year, with tree species, I tell people the resistance that I develop has to last, I say, at least 100 years. And I said, yeah, really, that's not true. It has to last forever. So is it durable? Well, it lasts forever. And then is it stable over environments? You might find that, hey, good news, bad news, we've got good resistance, it's generally stable, but maybe porterford cedar, if we plant in real wet environments, it won't hold up, or maybe in real droughty environments, it won't hold up. So you have to characterize the resistance. Phase two, after you determine whether there is resistance and answer to those basic questions, and they fill in your models, how bad it's going to be, and if you decide, man, it's going to get bad, we want to be able to restore certain areas, we want resistant material, then how do you go forth? And at that stage, you decide whether breeding is necessary and selection. Other things that will come up if you decide to go into resistance is who or what group oversees the resistant seed development, okay? And whether it's a co-op, NGO, the federal government, the state, how is that organized? And that will be important. When will resistant seed be available? How to maximize restoration, that's something that is species dependent. And I know in talking to Phil Cannon and others here, that's a prickly thing that you're all going to have to work out. Some of these ʻŌhiʻa sites are really harsh sites, so how do you actually restore and plant seedlings? What will it take to, if you go forth with ʻŌhiʻa and resistance, you're going to have a few people who are really passionate and drive this. You're going to have the public support, a sense of urgency, review of other successful programs. You don't want to create any of the mistakes that have been made before. You may make new mistakes, but that's okay. And you really want to jumpstart the program and get it going. Proper structure and staffing, and especially if a species so important as ʻŌhiʻa, if you end up having to go to a resistance program, you want that razor focus, you want reviews of the program, and you want that program really to move. And then continuity of funding and personnel, maybe a series of 5-year plans so you keep the focus. So that's kind of an overview of resistance. The people coming after me are going to talk about how directly what might go on with ʻŌhiʻa, both at kind of the research stage and that phase 1, and then if you do decide that there's a need for breeding, how to gear up for that.
Just as a little primer for maybe some of us that don't know what ʻŌhiʻa we have here, we have 5 varieties. ʻhiʻa is found kind of at the lower elevations on the newer lava substrates. Glabrema is kind of in the middle. It doesn't have hairy leaves, and it likes kind of more of the wet environments. And it's known as a late successional variety that overtakes ʻhiʻa on these older substrates. We also have Macrophylla, which is found in the same location as Glabrema. The only difference is that the bark is papery or flaky. Elizabeth Stacy is a very big proponent of merging the two, and it makes reviewing papers really difficult for me because, of course, the old literature says that there's 5. So we also have ʻŌhiʻa, which is a riparian variety. It's also mostly glabrous, but it's found only along the rivers in Hilo and Hamakua, which also makes it very difficult for seed collecting. Like JB said, there's not very many around. And lastly, variety polymorpha, which is found at the higher elevations, roughly 1,400 meters and up. It's also pubescent. And so yeah, there's this big gradient of polymorpha at the top to Glabrema in the middle and ʻhiʻa at the bottom. So based on these varieties, for my work in my master's degree, I focused on 4 of the more well-established varieties of ʻŌhiʻa, ʻhiʻa, Glabrema, ʻŌhiʻa, and polymorpha that are found on our island, and inoculating them with an isolate of Stratocystis leucoʻŌhiʻa in our lab. And from that measurement, I was able to calculate disease severity, which for the purpose of this experiment is generally the percentage of the plant or the ratio of the plant that is stained. And so roughly, we tested 13 ʻŌhiʻas, 35 ʻhiʻas, 36 Glabremas, and 38 polymorphas. As you can see, all the variety polymorphas died. All 38 died as 100% mortality. However, with ʻŌhiʻa, ʻhiʻa, and Glabrema, we did see some survivors emerge after the 17 weeks. ʻhiʻa seems to be the most promising, having the most amount of survivors come out of it. We'll start with the mortality on the left-hand side of the graph. Like in the previous slide, we see that polymorpha had 100% mortality, followed by Glabrema, and lastly ʻhiʻa and ʻŌhiʻa had some of the least amount of mortality, around 70 to 80%. Then looking at the mean number of days to death, it's interesting to note that the 3 varieties on the left, Glabrema, ʻhiʻa, and ʻŌhiʻa, all had about the same amount of time on average till they died, whereas polymorpha, the one that died 100% of the time, died the quickest. At least that's what we found in our growth chambers. Looking also at pretty much the staining inside the plants, we've also seen that polymorpha and Glabrema show the highest amount of staining within them. We also recently received some seed from Kauai of variety Dideri and also Waialeale, and our hope is hopefully by next summer to inoculate those. They're still really young. This is a picture of what they look like right now. They're very tiny. It usually takes about 1.5 to 2 years from seed to be able to inoculate and get the size we want to wound the plant without killing it right off the bat. We also are working on other Metrosideros species in the Pacific. We have tested already Metrosideros excelsa chromatosensis hybrids and Metrosideros kalina variety Tahiti red in the lab. And if I believe correctly, Lisa, the excelsa chromatosensis hybrids were not susceptible, but the Metrosideros kalina was susceptible to disease. This is what the excelsa look like now. They're about 6 inches to a foot, and they are getting really close to the size that we can use. Real quick, real brief summary of what I kind of went over. There may be some resistance out there based on what we saw screening the different varieties of ʻŌhiʻa on Hawaii Island. However, further testing of the survivor plants need to be done, and of course, further testing of other populations of ʻŌhiʻa on our island and other islands need to be done. We don't just stop at 38 plants of each variety. We got to keep going, and that's where a big part of this moves on to the rod resistance project that Christian's going to talk about next, moving into screening a larger number of plants and figuring out more of what's the minimum concentration we need to inoculate a plant and not overdose with spores.
Kainana and I drew straws, and I pulled the long straw. And so this working group is intending to build on the great work that Lisa and Blaine and others have pioneered in ʻŌhiʻa disease resistance, as well as bringing in the expertise that we have with Region 5 and Region 6. And so this expanded project, the goal is to go out and find these rod-resistant individuals. Obviously, another need is for funding. We have secured some funding from USDA Forest Service through the Science Technology Development Program, from PSW in Region 5, and from DOFA. The approach is to continue with some of the work that Blaine presented, expanding our rod resistance screening to begin including some of the features that Rich presented. So growing this preliminary effort into a resistance program. The methods are to go into the field and collect cuttings of various ʻŌhiʻa varieties from Hilo and Puna using a paired site approach. So one of our ideas is that we will look for individuals that persist in otherwise heavily hit stands with the idea that they may be survivors, that they have some resistance. But we're also going to pair that sampling with adjacent stands that don't have much ʻŌhiʻa death in them, that they have overall maintained some integrity as a stand. The idea being on a landscape scale, these heavily hit stands may be genetically related individuals that overall are sensitive to rod, whereas adjacent areas that are more intact, they represent related individuals that may be more resistant. We're also pairing this cutting approach with seedlings that are from seed, and we're collaborating with the Lion Arboretum to structure our sampling of seed from across the state, as well as with Nick Dudley, who's going to be coordinating a major collection for Maui to look at disease resistance. The methods are pretty straightforward in terms of actually getting into it, propagating huge numbers of trees. We're not going to be in the thousands, the way that Rich was able to do in Oregon at Dorena. We'll be in the hundreds, but we're hoping to look at hundreds of individuals across the state. And then inoculation trials at PBARC. The last piece of the approach is to outplant the most resistant and also the most susceptible genotypes, and to do field inoculation trials to verify what the lab inoculation results show. If needed, after all of this, in years to come, the potential is there to build a disease resistance program with the resistant individuals that we find. Outcomes, we hope to have an improved understanding of rod resistance in Metrosideros. As Rich said, it's not that straightforward to even understand what resistance means at this point for ʻŌhiʻa. It could be resistance to the disease moving through the tree. It could be resistance conferred by a reduced sensitivity to wounding, or it could be the way that the tree is formed, the way it grows in terms of branchiness and having crotches that accumulate the pathogen, and then during wind or storms, those crotches break and allow the disease to enter in. So there are a lot of potential reasons why a tree might be resistant. And then, of course, weeding out the climatic drivers of resistance, or the soil drivers or the age drivers that aren't really resistance but happen to be a condition of the site, making them more escapes than survivors. Some other outcomes, we are looking to operationalize methods of ʻŌhiʻa propagation, so going from hundreds or even many hundreds of individuals to being able to propagate many thousands of individuals and quickly. Operationalizing methods for lab and field screening of ʻŌhiʻa, the inoculation process, and then operationalizing methods for testing ʻŌhiʻa for rod resistance. And ideally, we create a supply of ʻŌhiʻa for large-scale planting and restoration, landscaping, and for biocultural applications. So that's it. Thank you.